our first presenter is going to be Dr. Carl Bradley. Dr. Carl Bradley is an extension plant pathologist and faculty member at the University of Kentucky. Carl received his PhD from the University of Illinois and has had faculty opportunities at North Dakota State University and the University of Illinois. His current research and extension program is focused on disease management in small grain crops and in soybean. And today he's going to give us a history of fungicide registration and kind of set the stage of where we're at now in the line of research that has got to this point. So with that, Carl, I'll turn it over to you.
All right, thank you so much, Andrew, and thank you everybody for attending today's event. We appreciate your attendance. As Andrew said, this is going to be recorded, so tell your friends about it if they weren't able to make the live version. So I'm just going to talk a little bit about a little bit of history of fungicide registration for management of Fusarium head blight in the U.S. And of course, you can't start a presentation on Fusarium head blight without showing some pictures. So you can see in the background some nice pictures of Fusarium head blight, the symptoms that you see there. So also before I begin, I just want to give you a few important fungicide resources. First off, the label. As we know, the label is the law. That's where you're going to find the most up-to-date and accurate information. So be sure you're reading the label before you make applications. Also on the Crop Protection Network, and you can see the website there, there's lots of good information. One of the things that would specifically be very important for fungicides is the Fungicide Efficacy Guide for wheat diseases that's updated every year. And the new version will be updated pretty soon, and will be on that website. And then Dr. Marty Chilvers later today is going to talk a little bit about fungicide resistance. But one of the resources that you can find about fungicides and especially fungicide resistance is the Fungicide Resistance Action Committee, also known as the FRAC. Their website is listed there, so there's a lot of good information there as well. So I just kind of have this divided up into some different time periods. Prior to 1998, that was sort of the dark times. At that point in time, there were really no fungicides registered for management of Fusarium head blight in the U.S. Around that time, European researchers had identified tebuconazole, which at that time, the only product that included tebuconazole was a product known as Folicure. They identified that as one of the more effective products for Fusarium head blight management. And around that time, some Section 18 emergency exemptions were requested by some different states for the use of Folicure for Fusarium head blight management, but those were denied by the US EPA because we needed local data at that time. Also, before I go too far, I should mention that a lot of this information that I'm presenting today came from some information that myself and Dr. Marcia McMullen, who used to be the extension plant pathologist at North Dakota State University, we presented this at a Fusarium head blight forum in Indianapolis in 2008. So you could go to the proceedings of that and find some of this information as well. So then in 1998, that's when the U.S. Wheat and Barley Scab Initiative kind of formed the uniform fungicide trials. That's when those were initiated, where lots of different states were involved in testing different fungicides. They were all tested uniformly across the different states using the same protocol, but looking at different wheat classes and different geographies. And Dr. Pierce Paul will be on the program later today as well and talk a lot about data that came from those trials. But that sort of initiated the ability to get data from the U.S. to start requesting Section 18 emergency exemptions and maybe moving the way forward to get full Section 3 registrations for some of these products. So from 1998 to 2007, after there was some local data, that's when annual Section 18 emergency exemptions were requested by State Department of Agriculture from different states for the use of Folicure. Those mostly were granted, I believe, and so that's almost a 10-year period where states were really relying on Section 18 emergency exemptions to manage Fusarium head blight with fungicides. During that period, the EPA had a special review of triazole fungicides, which would include tebuconazole, which is Folicure, and full Section 3 registrations could not occur during that time because of that special review. Finally, in 2007, that's when we actually received the very first Section 3 full registration of a product for management of fusarium head blight, and that product was Proline, which contains prothioconazole. Then in 2008, our toolbox was starting to get kind of full of different products that were available. Carumba, which is metconazole, was registered. Finally, tebuconazole as Folicure, and then later on, other products that contains tebuconazole were registered. And Prosaro, which contains prothioconazole and tebuconazole, was registered. And then 2018, a little more recent, we got Miravis Ace, which is a combination of propiconazole and pidoflumethafen. And then more recently, 2021, Spherix, which contains metconazole and prothioconazole, was registered. And then 2022, Prosaro Pro was registered, which contains a three-way combination of prothioconazole, tebuconazole, and fluopyram. So this table just shows all those products that were registered. One of the things I want to point out here, as you look at these different products, you can see the different active ingredients that are included within each of these products. At the top are the different chemistry classes. So we really only have two chemistry classes, the SDHIs and the triazoles. And you'll notice that there's a triazole in every one of these products. So really, we're very heavy on the triazoles and what we're using here. But we do have some SDHIs that are registered as well now. So these two that are in red, those are the most recent products that were registered. So just a little bit of a comparison. Prosaro Pro is the one that's probably eventually going to replace Prosaro. You can see that they both contain the triazole, prothioconazole, and tebuconazole at different rates, but Prosaro Pro also contains an SDHI. And then the other one is Spherix, and you can kind of see its predecessor, Carumba. They both contain metconazole. Spherix contains a little bit less metconazole, but adds prothioconazole to it. So that's pretty much the story on what we have available now. Appreciate your attention, and I'm going to pass it back over to Andrew.
Thank you, Carl. I appreciate that. And really the big point of like what Carl's bringing to the table is just to make an awareness for, how far we've come with fungicide management. And then he set a really good stage on that. If you have any specific questions with that, go ahead and throw them up in the Q&A box. There's none at this time, but certainly if there's something that comes up, we can address those at the end of the Scabinar. Dr. Bradley mentioned the U.S. wheat and barley uniform fungicide testing efforts, and I think this is a good segue for our next presenter. So our next presenter is going to be Dr. Pierce Paul. He is a professor and associate chair in the Department of Plant Pathology at The Ohio State University. He has research and extension responsibilities for diseases of field crops, and where his primary areas of expertise are in epidemiology and integrated management of diseases and mycotoxins in corn and wheat. And largely, he has been what I would call our gatekeeper of a lot of our data on the fungicide effort. And what we've asked him to do today is to give us a review of all the years of efficacy and timing data, specifically on wheat and what we have found out over the years. So Pierce, I'll let you start sharing your screen, and I'll let you take it from here.
Thank you, Andrew, and thanks to the organizers for giving me the opportunity to share some of the work we've been doing over the last several years on fusarium head blight and the use of fungicides. And Carl, you couldn't do a better job of leading this off very nicely. So you're going to make it easy for me to transition into some of the efficacy work we've been doing. So what I want to do today is talk about efficacy of some of the fungicides that Carl listed in terms of fusarium head blight control and Don. And like Andrew said, we've been doing this for several years. We've got hundreds and hundreds of data, but I'm going to really synthesize this very nicely in terms of some of the analysis we've been doing. We're going to look at products and look at application timing and talk a little bit about 1 versus 2 treatment programs in terms of reducing Fusarium head blight and Don. So like Carol said, it's hard to talk about Fusarium head blight without at least putting up a picture here. This is very typical symptoms of the disease. On the left, you've got a spike that's affected by Fusarium head blight, typical bleach discoloration of the spike. On the right, you've got a healthy spike at that same growth state. So this is clearly not maturity, is Fusarium head blight affecting the spike. And then in the middle, you've got a picture of a field that's affected. In terms of the consequences, this is fairly well known to everybody who's had to deal with Fusarium head blight. It reduces yield and grain quality, contaminated grain with mycotoxin, food safety concerns for human and animals if contaminated grain is consumed, and then it affects milling and baking quality of flour. Those are the overall effects of the disease. Carl mentioned this as well. We, as a group of wheat pathologists, we normally publish this fungicide efficacy chart. This is not a complete list of all the fungicides that are available, but it's a list of some of the most widely used fungicides, and we've got 20 on this list. And of the 20, we've got 8 that we consider to be scab products. We consider these to be scab products based on efficacy and also based on the fact that some of the other products that are on this list contain QOIs, which we typically don't recommend for Fusarium head blight control because we've got data showing that they are associated with increased levels of vomitoxin. So we've got 8 fungicides that we consider to be scab products. And of these scab products, we really do have represented 4 active ingredients. And again, Carl talked about this: tebuconazole, propiconazole, prothioconazole, and metconazole. So these 8 products, if you don't consider the generics of Folicure and the generics of Tilt, these 8 products are basically built around 4 triazoles or DMIs. And then we've got a couple of new active ingredients in the SDHI group added to our toolkit in terms of fungicides for Fusarium head blight control. So I'm going to share data on all of these products, and I'm going to focus mostly on the last 8 years or so of research we've been doing, because like I said, we've got a whole lot of data, and it's pretentious of me to think I can share all of that data in 20 minutes. So I'm going to start looking at Miravis ACE and with some comparisons of Miravis ACE with what I consider to be the industry standards, Pizarro and Caramba. And I'm focusing on Miravis ACE here first because there are lots of questions about timing of Miravis ACE, because part of the marketing strategy for this product was focus on using it at an early application time. So I'm going to focus on Miravis ACE at a couple of different application times, and then compare it to Pizarro and Caramba, and then compare it in 2 treatment programs as well. So the standard application time for fungicide for Fusarium head blight control is fixed 10.5.1. But with Miravis ACE, there was potentially an opportunity to apply this fungicides earlier at fixed 10.3. This is when 50% of the head is out of the leaf sheath of the flag leaf. So this is fixed growth stage 10.3. And we were excited about this possibility because this would give growers some flexibility in terms of timing of fungicide to control Fusarium head blight. Like I said, the recommended time is anthesis, when there is anthers sticking out the central portion of the spike. That's early anthesis. This is the standard recommendation time. But then we wanted to see, okay, we've tested before with other fungicides, and the early application didn't do as well as the anthesis application. But since Miravis ACE has a different active ingredient in it, we wanted to see, okay, are we getting some good residual effects that would allow this fungicide to be applied early and still provide adequate control of Fusarium head blight and DON. So here's a list of treatments from one of our fungicide efficacy trial, and this is more than 50 different trials, location years of data. And we've got the check, Pizarro anthesis, Caramba anthesis. Here are the 3 timings of Miravis ACE at heading at 10.3 growth stage, Miravis ACE at anthesis 10.5.1, Miravis ACE about 4 to 6 days after anthesis, and then some what we call sequential application treatments or combination treatments with Miravis ACE at anthesis and either Pizarro, Caramba, or tebuconazole 4 to 6 days after anthesis. And here I'm showing percent control. So this is not actual levels of scab or Fusarium head blight. It's percent control. This is percent reduction of the disease relative to the disease level in the check. And this is average across those trials that I talked about. And we've got Miravis ACE here, and everything, the center line shows 50% control. Everything on the right of the center line is more than 50% control. So essentially, all of these fungicides and all of these different application timing reduce the disease by more than 50%. If we look at products, Prosaro reduced the disease by about 60%, compared to Caramba, just about 58 to 60% as well. But look at Miravis ACE here. Miravis ACE at anthesis reduced the disease by about 70%. This is comparing these three products at the same growth stage. Miravis ACE at anthesis, Prosaro at anthesis, Caramba at anthesis. If we look at timing now, here you've got Miravis ACE at that 10.3 growth stage, Miravis ACE at anthesis, and Miravis ACE 4 to 6 days after anthesis. Clearly here, this Miravis ACE at the anthesis growth stage, close to about 70% reduction of the disease, compared to about 60% reduction with that early application. In other words, the early application did well in terms of reducing it, at least to levels comparable to Prosaro and Caramba. The late application also did well at about 60% control, but neither of those two applications did as well as the anthesis application of Caramba. Sorry, the anthesis application of Miravis ACE. So clearly, Miravis ACE applied at anthesis did better than Prosaro and Caramba, and it did better than the early or late application of Miravis ACE. At the bottom here, we've got the two treatment programs. All of these two treatment programs reduce the disease level, percent of the spike area damaged by the disease by more than 70%. If we look at DON, we see pretty much the same trend. Comparing products again, Miravis ACE reduced DON by about 50% compared to about 40 to 45% with Caramba and Prosaro. And then again, the early application timing did not do as well. It did fairly well in terms of reducing visual symptoms of the disease, but it did not do as well in terms of reducing the level of mycotoxin contamination of grain, just about 30% reduction compared to 50% with the anthesis application of Miravis ACE and about 50% also with the late application or application 3 to 6 days after anthesis. If you look at the combination treatments, they all reduce DON level by 60 to 65 or 70% with the double application of Miravis ACE at anthesis, followed by Prosaro, Caramba, or Folicure 4 to 6 days after. Shift gears a little bit here now and talk about some of the other new fungicides that Carl mentioned. Prosaro Pro, Spirex, and Miravis ACE, I consider these new fungicides. And again, compare them against the older fungicides, which are likely going to be replaced by some of these newer fungicides, and then look at double applications again as well. So here's our list of treatments. We've got Prosaro, Caramba, Miravis ACE, Prosaro Pro, and Spirex at anthesis. And then the two application treatments here include Miravis at anthesis, followed by Prosaro Pro 4 to 6 days after anthesis, Spirex 4 to 6 days after anthesis, and Tebuconazole 4 to 6 days after anthesis. These are the two treatment programs. I've highlighted a couple of programs here for reasons mentioned by Carl. Prosaro Pro is probably going to replace Prosaro, so it makes sense to want to compare them to see if this is just as effective or more effective. And then Caramba is likely going to be replaced by Spirex. So again, it makes sense to try to compare these two to see how effective they are relative to each other. So here's some data I'm showing you here. This is not percent control this time. This is percent scab. This is percent spike area covered with symptoms of the disease. The check has about 11% scab. And each of these fungicides, based on the letters here, can tell you that each of these fungicides resulted in significantly lower average level of scab compared to the check. So they were all effective against Fusarium head blight. They are all effective against visual symptoms, resulting in significant reduction in the level of disease. However, the efficacy varies with fungicide, and we're going to look at efficacy in terms of percent control. The red box here shows Miravis ACE. It reduced the level of disease by 76% relative to the level of disease in the check. So if you look at this level of reduction here is 70% lower than the level of disease in the check, compared to 58% for Ferrex, 60% for Prosaro Pro, 55% for Prosaro, and 37% for Caramba. So clearly, Miravis ACE is the most effective of these single application treatments that we looked at. For the double application treatments, the efficacy ranged from 58 to 74%, with Miravis ACE at antithesis, followed by Prosaro Pro, and Miravis ACE at antithesis, followed by Spherex, resulting in about 70 to 74% reduction in the overall level of disease. So clearly, the double application program performed better than most of the single application programs. Just to break this out a little bit, side-by-side comparison of Spherex and Caramba. Spherex reduced the level of disease by 58% compared to 37% with Caramba. So clearly, Spherex is more effective than Caramba at reducing the disease. And similarly, the comparison with Prosaro Pro and Prosaro, they are of comparable efficacy. Slightly better results here in terms of percent reduction, but statistically, these are not significantly different. So Prosaro Pro performed comparably to Prosaro in terms of percent reduction of visual symptoms of the disease. If we look at DON, we see pretty much very similar trends. The overall level of reduction relative to the check is much lower for DON compared to visual symptoms. But again, we've got 6% scab, 6% DON in the check, and every single one of the fungicide treatments we evaluated reduced disease, reduced mycotoxin compared to the check. Here again, Miravis ACE resulted in 53% reduction of the mycotoxin contamination of the grain, compared to 47% for Prosaro Pro, 41% for Prosaro, 40% for Spherex, and 23% for Caramba. Following the same trend here, the double application resulted in 46 to 66% reduction, with Miravis ACE followed by Spherex and Miravis ACE followed by Prosaro Pro, resulting in close to about 65% reduction in the level of the mycotoxins. So clearly, the two application treatments performed better than the single application treatments in terms of reducing the overall level of mycotoxin contamination of grain. But of the single application treatments, Miravis ACE stood out again as the most effective. Again, side by side, Caramba is less effective than Spherex in terms of reducing mycotoxin contamination of the grain. And then Prosaro Pro and Prosaro are of similar efficacy in terms of reducing the level of mycotoxin in the grain. Prosaro Pro is a little better than Prosaro, but again, statistically, they're not significantly different from each other in terms of their efficacy at reducing the level of mycotoxin in the grain. So some quick take-home messages. Miravis ACE at antithesis provided the best single application control of Fusarium head blight and DON. Early application of Miravis ACE was less effective against the mycotoxin. It did fairly well at reducing visual symptoms, but it was less effective against DON compared to the antithesis application, and that application made shortly after antithesis. Prosaro Pro and Spherex were of comparable efficacy to Prosaro. Spherex was more effective than Caramba. And then in general, the two application programs led to the highest level of disease control and mycotoxin control in general. But this has to be looked at from an economic standpoint, and that's part of the future work we're going to do, is to see what's the cost benefit of doing some of these two application programs. Before I finish up, I want to acknowledge my group. I've had several postdocs over the years work on this disease. Wanderson and Marion were two recent postdocs. Isaac is a new postdoc. and my graduate student, Jorge, he's working on Fusarium head blight as well. And I'd be remiss if I don't thank everybody who's been involved in this project, a long list of PIs across the different wheat growing regions who've contributed to the project. And that's all I've got. Here's my contact information. Feel free to contact me if you got any questions. Thank you.
Thank you, Pierce. That was a really nice review of, it's a lot of data, right? We always joke
It's a
about it. It's only
lot
like,
of data.
yeah,
I
a
like,
couple
you know, I
slides.
like it that way, right? But yes.
That's good. So Pierce, we have a little bit of time here.
Okay.
Wondering if you could answer a question or two. The first one that popped up on my screen is, I think this was referenced to one of your earlier slides, but a question was, how much Dawn was there in the untreated treatment? My guess is when you're showing percent control, I guess the curiosity was, my guess it depends. There's a wide range.
Right. That's a wide range. And yes, like I said, that's meta-analysis. It's a good way to standardize data across trials, all the variations in trials, is by looking at it in terms of percent control. But I think the Dawn levels were about 8 parts per million in that average across all of those trials. It's multiple years of data.
Okay, thank you. The other question we have, this might be a great topic to bring up later too, and you could provide some comments right now, Pierce, but how would you recommend timing a fungicide application to growers that experience long period of anthesis? I'm guessing it's uneven growth stages, right? We always get this question, but I don't know if you have any comments right now, and we can address this as a group at the end too.
Yep. Yeah, I can just briefly touch on that, but I'd love to hear I'd love to leave it for the group discussion as well. I think that's always a challenge, and we don't have that very often in Ohio, but in years where the grain field period is cool, anthesis is extended. And those might be years where I think some of those two treatment program, double application program might make sense, because you get a larger swath of those spikes at that anthesis growth stage. If you do that 2 application program, start with the earliest flowering material, and then come back in a couple of days late. Obviously, the economics is going to be a big deal there, but then you've got some of these fungicides that are off patent, that are fairly cheap, that might be good options for the second application.
Yeah, that's a good discussion point. I think we might bring this up later too, because different market classes might be some different ways.
Absolutely.
Well, Pierce, there's some more questions rolling in. If you have time, you can respond to them via text and in the box. But I thank you for your time today. And if you could stop sharing your screen, we can keep moving along with the program. So our next topic that we're going to be moving to is, Pierce did a nice job on the wheat side. Now we're going to touch on the barley side. So what we have as our presenter for this first part is Dr. Christina Cowger. She is a research plant pathologist with the USDA ARS and based in Raleigh, North Carolina, and is also an adjunct professor in the Department of Entomology and Plant Pathology at North Carolina State University. Her primary research focus is understanding and managing economically important diseases of wheat and barley. And what we have asked her today is to review some of the efficacy and timing work that she's conducted on winter barley. And then I'll just provide a few slides at the end on some spring barley for comparisons. But most of it, I'm going to hand it over to Christina, and your screen looks great, and you can roll from here.
Great. Thank you very much, Andrew. And it's great to be here today to talk with you about, excuse me, about Fusarium head blight in winter barley, specifically about fungicides and the timing of application of fungicides for control of scab. And this is an area where, in contrast to the data-rich environment that Pierce was just talking to us about, we haven't had a great deal of replicated research. So we've been attempting to remedy that gap by studying the timing for best management of FHB in winter barley. Where we are in North Carolina, winter barley is the crop. So for those familiar or unfamiliar with the symptoms of FHB in barley, on the left here we've got 6-row barleys, which are mostly used for feed, although some of them go for malting as well. And you can see the brown kernels that have emerged on these heads of 6-row barley. And then on the right, we've got 2-row barley, which is almost entirely used for malting purposes. And you can see the symptoms in that head as well. Now, unlike wheat, barley generally does not allow the spread of fusarium fungus within the spike. So in wheat, the fungus gets into the spike and it travels up and down the spike via the rachis, or that central stem. But in barley, that's not usually the case. Now, it is true, however, that in 6-row barley, the fusarium can actually move directly from floret to floret, from kernel to kernel, outside the rachis. So you can get movement up and down the head in that way. And here on the right, we're looking at some barley kernels that are affected by scab. Excuse me. And you see some pink discoloration and then some little black spots, which are the fruiting bodies, perithecia, that through which the fungus reproduces. So what about the tolerance of our markets for DON, deoxynivalenol, in barley? Well, I'm going to focus here on malting barley because that's where tolerances are the lowest. If DON is greater than about 1 part per million, load is likely to be rejected for malting purposes. And why is that? Well, maltsters appreciate that DON could be transferred from the malt into the beer, so that's one concern. And surveys have shown that DON is commonly present in beer, but at extremely low levels. So it's not really a health hazard, but of course, nobody wants to think that there's deoxynivalenol or vomitoxin in their beer. And so public perception is important there. What other concerns are there about FHB in malt barley? Well, fusarium, which remains alive in the harvested grain, can grow again during the malting when moisture is added. And that can lead to additional production of DON in the malt. Here on the left, you see fusarium on these purplish kernels in germinating barley in the malting process. So that's one problem. You can get more DON while the malting is going on. Then also, fusarium could damage the components of the grain that make up malting quality. And that's no good because, of course, malting barley is all about the malting quality. And then finally, fusarium can produce chemicals when it metabolizes that cause gushing in beer. And so you can get, when you open a bottle of beer, you can get the beer gushing out. And naturally, that's very undesirable in beer. Feed barley also has limits on DON. And I won't go into that in detail, but 5 parts per million for swine and 10 parts per million for other livestock are generally the limits in wheat grain and presumably also in barley that's used for feed. Okay, so we want to protect our barley from fusarium infection. When is it susceptible? When should we be thinking about that? Well, in North America, winter barley generally flowers while it's still in the boot. So here in the lower left, you see some stems with boots getting ready to swell with those heads that are about to emerge. And that seems to be often, at least, where the flowering process actually takes place before the head ever emerges from the boot. And so when heads come out, you usually don't see extruded anthers, at least in the mid-Atlantic region where we do our research. It's rare to see anthers coming out of those heads. Now, spring barley is a different story. It often extrudes its anthers, as you see in the lower right here. And so maybe fusarium is infecting at that point when the anthers are coming out of the spring barley heads. The truth is we don't really understand very well when those barley heads are vulnerable to infection by fusarium, like we understand in wheat, where the extruded anthers are the infection court. We don't really understand that for barley. And so on that basis, we wanted to conduct our investigation of the best timing for protection of barley with the fungicide. And so here are some possible maturity states, as Pierce has just discussed. Well, with barley, you've got on tipping, 50% emergence of the heads, 100% of the heads just emerged, and then about 6 days later. So those are the timings that we chose for our 4-year experiment in an inoculated misted nursery in North Carolina. So we did 4 years of work on winter barleys. We had 3 timings: the early, which is 50% heads emerged; the mid, where heads are just 100% emerged; and then the late, 6 days after 100% head emergence. And you can see a significant amount of peduncle has emerged, which helps you distinguish visually what that late timing looks like. And then, of course, we had an unsprayed control. Now, this experiment involved 3 fungicides, and we've already heard a good deal about these products. They were Prosaro, Caramba, and Miravis Ace. And so we've got our triazole chemistries in there, and then the SDHI with Miravis Ace, the newest product of these 3. But 3 good products for control of FHB in wheat. We used 3 winter barley varieties. One of them, a 6-row variety called Thoroughbred, which is widely popular and used both for feed and for malting. And then 2 popular 2-row malting barley varieties called Violetta and Flavia. Okay, so what did we find? Well, here's some index results. These are symptoms, basically the percent of kernels that are symptomatic, like this. And so you see that the non-treated treatment, and this is averaging across varieties and fungicides and years. Non-treated, we had about 6.5%. The early timing, about 3.5%. Mid, about a little less than 3, and late, 2.5. So basically, every fungicide timing was better than the non-treated control, but early was significantly worse than either the mid timing, which is 100% heads just emerged, or that late timing 6 days later. Those were the best for controlling symptoms, the mid and the late. Better than that early timing. What about dawn? So here, the blue bars are dawn, and dawn goes from 0 up to 3.5 parts per million here. So our non-treated mean, and again, this is averaging across fungicides, years, and fungicides, years, and varieties. Non-treated, just a little below 2 was our average, 2 parts per million. Early around 1.5, mid around 1.3, and late was the only timing that got us under that 1 part per million of dawn where we're trying to get with malting barley. So late was significantly better for reducing dawn than either mid or early, and early was really no different from the non-treated control. These orange bars are percent infected kernels. So we plate these scabby kernels out and see how many of them are actually infected with Fusarium. And there we got a similar pattern, really, where in this case, the early fungicide application was better than the non-treated control. These are our orange bars. The mid timing was better than early, and the late timing was the best of all at reducing percent infected kernels. And that's significant, of course, because the maltster doesn't want those infected kernels, which could start growing Fusarium again in the malting process. So late timing better than mid, mid timing better than early, and early for dawn control no different from the non-treated. Similar result to what Pierce was showing us. Now let's look at those fungicide products briefly here for both the visual symptoms and dawn. So this is our visual symptom axis over here on the left. And you can see that in this case, it's the blue bars, that Caramba was provided better reduction of symptoms than the non-treated plots, but Prosaro was better than that, and Miravis was best of all. For dawn, the orange bars, we've got Caramba, in this case, actually no better than the non-treated treatment, non-sprayed treatment, and Prosaro and Miravis Ace, both superior at controlling dawn. So this also accords with Pierce's results for wheat. So Miravis Ace, Prosaro doing better at dawn reduction than Caramba, and all three of the products better than the non-treated control. In summary, what we have concluded and what we're recommending for winter barley producers at this stage is for timing to wait a little bit after full head emergence and to let those peduncles elongate a little bit. And we seem to be getting better control of both our symptoms, but importantly, our seed infection rate and our dawn levels if we wait a few days, 6 days in this case, after full head emergence. Thanks. That's all I have today, and I'll turn it back over to Andrew.
Thanks, Christina. As I kind of mentioned beforehand, Christina did a really nice job of laying out more of the methodology that we've done over the years with the wheat and also in the barley. And what I wanted to get across is we have a similar complementary data set also in spring barley. And I'm just going to give you 3 slides because I think the message is kind of clear on some of the trends we've seen in wheat and barley. And again, I'm throwing up some of the growth stages up here. This is on some 2-row spring barley. And really, when I get asked the question about the timing in spring barley, this is what I'm resorting to. Our timing for spring barley starts at, in this case, full head emergence, and it extends for about 7 days. The one thing with experience in spring barley is, don't pull the trigger too early. When we start seeing the on emergence, I always tell folks to wait. And then they say, I still see it in 2 days. I'm like, just wait, because it goes back to fungicides, in this case, are only as good as what they cover. And I know Rob Prue is going to be touching on that in a little bit. So again, when it comes to a timing, really starting to see this back end of that 7-day window. And if you want data to support that, what I've done is I provided some summary data results that we've done in spring barley. Again, I took as many locations we have for spring barley and broke this down into a couple different treatments across multiple locations, multiple experiments. And really what I'm showing you is a non-treated control, so an unsprayed check. Prosaro at that full head stage, Miravis Ace, Prosaro Pro, and Spherix, all at that full head stage. And then that late application is what we've often termed it being 3 to 7 days later. And this first data set I'm showing you is the disease index. So as Pierce and Christina said, more of the visual symptoms and the plots. And here you're going to kind of see that story is that when you make that fungicide application beginning at full head, all of them perform fairly similarly from the last couple of years when it comes to spring barley. And that late application of that Spherix 3 to 7 days later, really doesn't matter if it would have been Prosaro, Miravis Ace, or Prosaro Pro. The same trend that we see in the data is we have 7 days to make an application in spring barley. Using the same mindset for the dawn analyses on this case, again, same treatment list, dawn levels in the non-treated check around 2 parts per million. You're going to see that 15-75% reduction depending on the treatment. But again, we do have some effective fungicides, and when applied at the right time, do help as far as managing of Fusarium head blight and dawn. So kind of merging what Christina and I wanted to do is what she's finding with that late application being the recommendation for the winter barley. We still see the value of that window and also that late application in spring barley instead, because that's often a common question we get is spring and winter barley recommendations. So I just wanted to kind of bring it full circle in that case. So Christina, I have you turn your camera back on if you don't mind. I have not seen any questions pop up yet, specifically for this presentation, but I encourage the group that if you do have any questions, specifically for really anything, looked like the question box is pretty active after Pierce's conversation. So that's good. So I encourage you to keep doing that. We have several panelists, several presenters that are willing to answer those questions. But Christina, I'd just like to thank you for your time and again, doing a lot of excellent research in North Carolina and solving some of these winter barley scab questions that you've been getting. Awesome. Well, we're going to keep moving along here. The next segment, the next presentation that we're going to have on the program, we have asked Dr. Rob Frew, who is an Extension Agriculture Technology Systems Specialist at North Dakota State University. And he assumed or began this role in 2023, but he has hit the ground running. And he has a lot of experience in the classroom lectures, has a lot of experience with GIS, experience with pesticide application technology. And we felt as a committee that he would be an excellent resource to give us some of the summaries of the fungicide application questions that we get in scab, coverage, water use, nozzles. And what Rob is tasked with today, he's going to give us a viewpoint of what the research suggests and give some comments and recommendations. So Rob, I'll turn it over to you.
All right, very good. Thank you. Great to be with you today to share a little bit on application technology as it relates to fungicide applications for FHB and DON management. One of the things that maybe sets me apart a little bit from some of the other panelists is that I don't truly have personal experience in wheat research and in FHB management in wheat research. So my master's work was in soybeans and my PhD work was in some crop modeling. So I don't really have that direct experience, but as Andrew mentioned, I'm fairly new into this role as an agriculture technology systems specialist. And a part of that responsibility is sprayer systems. And in the early stages of my work, I've been looking at some of the resources that are available from NDSU Extension and things that maybe haven't been updated in a while. And I found this particular publication on ground application of fungicide for suppression of FHB in small grains. And this was published by our extension service in November of 2008 and hasn't really been updated since then. So I've been looking at this and seeing what the more recent research is telling us compared to what's in this publication and working on an updated publication to come out of NDSU Extension. So what I'm going to share with you today is really the main points from that in progress publication that I'm currently working on. And that publication summarizes research from 18 different publications investigating such things as spray coverage or deposition on some sort of a vertical target. And sometimes this is actually on a small grain's head. Other times this is an artificial target, such as a plastic straw. Otherwise, these publications investigate things such as effect on grain yield, effect on disease severity as measured by FHB suppression or by the presence of DON in harvested grain. And this publication is mostly informed by research in North Dakota and Canada, seemed to be most applicable to a North Dakota perspective, but I think there's some lessons to be learned in other areas as well. So the first point I'll make from this publication, from all this research, is that the evidence suggests using a dual angled spray for ground applications with a steeper angled forward-facing spray, for example, 30 degrees from vertical, and a shallower backward-facing spray, for example, 70 degrees from vertical. And the angled component of this recommendation, the evidence for this is actually quite clear. Across all the 18 studies that I've looked at, the evidence was there that an angled spray always provided equivalent or better coverage of vertical targets compared to just a straight down spray. And when there was a benefit to this angled spray, coverage was increased by up to 15 to 74%. So the evidence is really clear to use an angled spray on a vertical target rather than just a straight down spray. The rest of the components of this recommendation, so why a dual angle and why a steeper angled forward-facing spray and a shallower backward-facing spray, really has to do with trying to get the most uniform coverage you can on that vertical target. And this graphic from the folks at Sprayers 101 illustrates this well, where if you use a conventional fan nozzle with a straight down spray, you'll get high coverage on the leading edge of the spike, but you'll get poor to no coverage on the trailing side. If you use a symmetrical twin fan type nozzle, you will get coverage on both sides of the spike, but your coverage on the back side or the trailing side of the spike is going to be low, because that spray has to overcome the momentum of that backward facing spray has to overcome the forward travel momentum of the sprayer. So if you tilt that rearward angle upward and create a shallower spray, that will better, the momentum of those droplets will better overcome the forward momentum of the sprayer, and you'll do a better job of depositing droplets on that back side or that trailing side of the spike. And you may not get quite as high a coverage on the leading edge of that spike, but you'll get a better overall, more uniform coverage of the entire spike, which seems to have a benefit for FHB suppression. And the benefit of these asymmetrical sprays with that shallower rearward facing spray is going to be more pronounced if you have faster ground speeds, for example, 12 to 15 miles per hour versus 6 miles per hour, combined with larger droplets, for example, a coarse spray quality versus a finer medium spray quality. And all of this is relevant for some of the conditions that we seem to encounter here in North Dakota. And this leads me to a second recommendation from this upcoming publication, is to recommend using a coarse spray quality, or what you might otherwise hear called droplet size. And this is a little bit of a change from our 08 publication, which recommended a fine to medium-sized drop of 300 to 350 microns. But I think what's important to realize is that when you talk about spray quality, that is actually a mixture of different droplet sizes. So for example, this is some experimental data showing the droplet size profile of this particular flat fan nozzle applying a fine spray quality. And it doesn't provide just a single droplet size, it provides a range of droplet sizes. Most of the volume is coming in droplets of about 150 to 200 or 250 microns, which in that jar of objects there would be the red, light blue, and the really small yellow ones, which are really kind of settled to the bottom and you can just barely see them. And those are all fairly drift-prone droplets. The ideal droplet size, that 300 to 350, is actually represented by those black beads in the container. And if we look at a droplet providing a coarse spray quality, there actually is a similar number of those in a coarse spray quality. It's just you have much fewer of the more drift-prone droplets of 150 microns or less, and instead you have some of the larger droplets represented by the golf balls or larger objects in this jar. And the reason why a coarse spray quality is recommended is that larger droplets have a greater mass. And that becomes really important because with a greater mass comes greater momentum or sustained movement towards the target, and also greater inertia or resistance to being moved off target by some sort of, or resistance to a change in motion moving that off target, for example, by wind currents. And this gets to be really important when you're trying to project a droplet to land on a vertical target. That momentum is really important, especially again for that rearward facing spray when you're trying to overcome the forward moving momentum of the sprayer. So having those larger droplets with a greater mass and a greater momentum really helps to carry them towards those spikes. And if you have faster ground speeds with more turbulence behind the boom, or if you have breezy conditions like what we have in North Dakota, that greater inertia becomes really important at keeping those droplets moving towards the target. Another benefit of larger droplets is that a complicating factor when making scab applications is that awns will catch droplets, but those larger droplets are going to have a better ability to move past the awns and land onto the glooms, and that's what you need in order to get FHB suppression. So that's another benefit of the little bit larger droplets of a coarse spray quality. Third recommendation is to use 10 to 20 gallons of water volume per acre. You're going to see a consistent coverage benefit to higher water volumes. The evidence is pretty clear on this. If 10 gallons per acre is going to be the acceptable minimum water volume, If you really want to optimize your coverage, you should be looking at something like 15 to 20 gallons per acre. I know in our area, this can be a tough sell sometimes just due to operational efficiencies of having lower spray volumes. If you're really concerned about operational efficiency, that 12 to 13 gallons per acre might be a little more of a reasonable compromise between the operational efficiency of hauling around less water versus the better coverage you're going to get with the higher water volumes. A fourth point is to keep that nozzle height as low as possible without dragging through the spikes. Ideally, you're going to want the nozzles to be 9 to 12 inches above the heads, and certainly no higher than 24 inches above the heads. If you have excessively high boom heights, you're going to see a decrease in coverage. Some experimental data that illustrates this nicely was done looking at a nozzle produced by TJet, an asymmetrical nozzle produced by TJet that's designed really for these types of applications on small grain heads. And with that particular nozzle, the nozzle height that's recommended is 15 inches above the spikes. If you raise that by just 5 inches up to 20 inches above the spikes, the spray deposit was reduced by 19%. And if that nozzle height were reduced or were increased up to 30 inches above the spikes, that deposit was reduced by 60% relative to the 15-inch height. So that nozzle height becomes really important for optimizing coverage. And finally, the last point I'll make today, summarizing up this research literature, is that ground speeds of up to 12 to 15 miles per hour are acceptable as long as you can maintain proper boom height, proper stability, and proper spray pattern. And this is something that I think there's a lot of local context to it. In areas that have small, irregular shaped fields, these types of ground speeds would be probably inconceivable. In North Dakota, we have a lot of large fields that are rectangular in shape, so we do have applicators that apply at these ground speeds. And it can work. You might see slightly less uniform deposit between the front and back of the head, but you should see no overall negative impact on coverage. Important point, this does assume that you're using a coarse spray quality. If you were using a fine or medium spray quality, then the turbulence behind the boom would move much more of that spray off target. And again, assuming or maintaining that stable boom height of 24 inches or less above the heads is really important. And that's something that becomes more and more difficult to do as you drive at higher ground speeds, to make sure you watch that boom stability and just otherwise pay attention to the turbulence behind the boom as well. If it seems that turbulence behind the boom is compromising your spray pattern, then by all means you should consider slowing down. So a couple overall points to kind of finish off my time here. It's really important to realize that you cannot isolate any one of these variables from the rest. This became pretty clear to me as I was going through the literature. And to illustrate this, I'll just give you a couple different nozzle configurations out there that seem to be highly successful in suppressing FHB. The first one is from some research done by Dave Hooker up in Canada. And he found that he optimized FHB suppression by using turbo flood jet nozzles alternating forward facing and backward facing along the boom at 20 gallons per acre water volume and 12 mile per hour ground speed. And those flood jet nozzles produce a pretty coarse spray, a really coarse spray, and ultra coarse spray quality to be exact. And this worked well, I suspect, because of the 20 gallons per acre water volume. As you get bigger droplets, you get poorer coverage, but you can offset that by using more water volume. But if you were using 10 to 15 gallons per acre and ultra coarse spray droplets, I suspect that you would get unacceptable coverage. So again, you can't look at these things in isolation. You have to consider the whole system together. Compare this to NDSU recommendations from that publication published in 2008, where it recommends a forward-facing flat fan nozzle, 10 gallons per acre. These applications were made at 6 mile per hour ground speed with fine to medium spray quality. And unfortunately, I don't know what the wind speeds were. It wasn't reported in any of the literature. And I suspect that part of why this system worked is due to the slower ground speeds of 6 miles per hour. And I'm guessing the wind speeds were probably low as well, just based on when researchers tend to implement their field trials, probably in the morning, probably during the calmest part of the day, not necessarily representative of when applicators are doing this work. And I suspect that if you had a fine to medium spray quality with breezy conditions like we get in North Dakota, or with faster ground speeds, that you would be seeing some more drift and less favorable results. So again, it's important to consider the entire system. And to relate this topic to the rest of the topics here in this webinar, realize that fungicide application settings are important for FHB suppression, but your product selection and application timing are going to have a greater influence. If you execute your fungicide application perfectly, that's not going to make up for a poor application timing, nor is it going to make up for selecting an ineffective fungicide product. So if that good fungicide product and the proper timing is, the ice cream sundae, then the application technology is the cherry on top. And with that, thank you for your attention.
That was great, Rob. Nice summary. And we gave you a tall task and, in 15 minutes to cover all these questions that, as plant pathologists, we sometimes are trying to figure out. So there's one question here, but, this might be more for the group, but I guess the question that, we have a group of experts behind the scenes, but there are lots of adjuvants out there with many recommendations. What properties should we keep in mind? Oil versus NIS, organosilicone, just more of the adjuvant as far as, so I mean, Rob, probably, I'm not sure if you have any comments on adjuvants or the different types of adjuvants and how they work in application, but
I don't really.
Yeah, that's fine. That's something we can punt to the end when it comes to our panel discussion. Other than that, I'm not seeing anything come in at this time, but I'd like to thank you, Rob. That was an excellent review of what's out there, and it definitely answers a lot of the commonly asked questions that folks will get from making an application. So thank you for your time on that. We're going to move forward here and just kind of give the group a heads up. We have one more presentation, and then we're going to take a very small break to set up our what we call a roundtable discussion aspect of it. And really what I'd like the audience to do is start thinking of those really tough questions, start submitting them in the Q&A, and then, there's going to be a panel of, I think, 6 or 7 of us that are going to unravel some of our mindset on that. So just kind of a heads up, we have a little bit of time, but if you have some good ones, make sure you use that question and answer box. So the final presentation we have today is we talked about fungicide efficacy and timing and application technology. But now we're going to move into a new effort that has been funded by the U.S. Wheat and Barley Scab Initiative. And this has to do with fungicide resistance monitoring and trying to develop some sensitivity. And with us today, we have Dr. Marty Chilvers, who's he's a full professor and field crop extension plant pathologist in the Department of Plant, Soil, and Microbial Sciences at Michigan State. And his research and extension program has a strong focus on minimizing losses caused by field crop diseases. So with that, Marty, I'll turn it over to you, and you can give us some perspective on the fungicide resistance monitoring effort.
Excellent. Thank you, Andrew, and good afternoon, everybody. Okay, so let's jump into it. So what I'm going to do today is present a little bit of work that one of my PhD students had done, and that sort of segues into what we're doing at the moment, which is sort of a more national view of fungicide sensitivity, just checking for fungicide resistance across all US wheat production. So let's just jump in then. So I'm going to focus primarily on Michigan data, but a lot of this applies to the national work we have going on. So just really quickly as a brief background then, of course, how does fungicide resistance develop? Well, it's through selection pressure. There's always going to be those few individuals in a population that will have some level of resistance. So as we make repeated application, we can skew that population towards resistance through repeated exposure. So just an important principle to understand. And we certainly have lots of examples of this. Within field crops, we're more or less limited to 3 primary groups of fungicides, main sort of modes of action there. So there's this group that Carl had mentioned before, the Fungicide Resistance Action Committee. So they get together annually and review data to help basically prolong the longevity of the tools that we have. And so they have a table and some information there about how these different fungicides sit and how they sort of group them out. So Pierce talked a lot about these demethylation inhibitors in terms of their action for suppressing scab. So did Christina, of course, in the barley. So that's one of the primary groups is group 3 fungicides, these DMIs. They target membrane synthesis, and they have a medium risk for resistance development. At least that's the sort of prediction that is out there. The other group that's pretty new, and we'll touch on this again, within the newer compounds or newer formulations that are available, also includes a group 7, such as the SDHI, or that is the SDHI group of fungicides. These target respiration, and they have a medium to high risk for fungicide resistance development. And I thought I'd just put in the strobilurins, or the QOIs. We don't use these for head scab management, but these are a good example of a class of fungicides that is at high risk for fungicide resistance development. And we've certainly seen that in a number of organisms that affect field crops. So frog-eye leaf spot of soybeans, Cercospora saginea, is a really good example of that. We've seen rampant resistance to that group of fungicides. So these fungicides are grouped in these different groups because of the target that they have on the organism and their various risks. So again, here's a list of products. As you'll see, especially the older products are all targeting that, or all of the group 3 fungicide group. So the DMI, so they're all basically targeting the same mechanism. And we have these couple of newer products now, Mirabis Ace, that includes a group 7 fungicide, and Prosaro Pro also includes a group 7 fungicide. So we're really reliant on a couple of, really just 2 modes of action for head scab management. So certainly, fungicide resistance is a concern. And so that's why we've initiated some of these various projects. So just quickly, I'm going to go through some of what we've done in Michigan to date. And now we're certainly broadening this out across all other acres. So what we did in Michigan is went out and collected a whole bunch of grain and head samples of wheat. The sampling was focused pretty heavily on wheat, but we also brought in corn, popcorn, soybean, and dry bean into the mix here, looking at the fusarium associated with those. Because when we look at when we isolate these cultures and look at them in the lab, they can be pretty hard to identify based on subtle differences in spore morphology and how they look as they grow on Petri plates. So we actually use some DNA techniques to identify and understand what species we're actually working with. So this is a really good breakdown for what we found in Michigan. So again, wheat, we had about 384 isolates that we went through and identified. The vast majority was certainly Fusarium graminearum, but there were a number of other species in here, and we sort of divide these into species complexes. You can see we've got a list of about 16 species or something here. The primary one, again, for wheat was graminearum, but we did have some other species, particularly in the Trisynctum complex, that appeared. You look into corn, and the different colors across those boxes just sort of represent the variation. Fusarium graminearum is certainly in there again, but then also this other species complex here, like subglutinins, was another species that was highly present in corn. So a lot more diversity in corn, but within wheat, it's not all graminearum, and that makes some of the work we do a little bit more complex, not targeting one pathogen per se, but there could be a complex of pathogens. So then, how do we assess for fungicide sensitivity? And I guess I just want to make a plug for all of the work that we do and all of that data that Christina, Pierce, and all of us are working to collect. These field trials that we do are really handy because they do give us a sense in field every year how these fungicides are working. Do we see failure in the field or not? And so there's tremendous value in continuing to do that work. But in the lab, we can put more discrete values on fungicide sensitivity or resistance. And the way we do that is we work with these fusarium cultures, And we'll basically create what we call poison plates. So we will mix in the fungicide product at various rates and then look at the amount of inhibition that has on that fusarium culture. So you can see this cartoon represents that from 0 parts per million of that fungicide product we're testing through to 30 parts per million. And there we can see the reduction in that growth. So we measure that growth. We run through some models to basically estimate the concentration that limits growth by 50%. So once we get down to 50% growth inhibition, that's how we would define the fungicide sensitivity. So in this case, we could maybe estimate that 1 parts per million was where we were seeing inhibition by 50% of that growth across the plates. So really briefly, here's what we found. So we found differences by the species in terms of the amount of fungicide needed to reduce growth by 50%. That Fusarium graminearum shown there in blue. And what we're really interested in here is the right-hand side of this graphic. So what you have here is the amount of growth at this fungicide concentration. And so we're looking really for those individuals that basically are not inhibited, or it takes a lot of fungicide product to inhibit growth. And that's what we would be concerned about. And really, some of the more important work here is to go through and assess where we are at and then check over time as well. These DMI fungicides, which is the primary class that we use, we tend to see more of a gradual shift in fungicide resistance development, as opposed to some of these other fungicides, maybe like the SDHIs, where you could see a single mutation that results in pretty significant shift in terms of resistance development. That's essentially what we're doing. What we took was to sort of do some in-field validation from this as well. Is took some of those isolates that we found to be less sensitive to the fungicide. So some of these isolates up here on the right-hand side of the graphic. And we put them out in the field. We sprayed them back out in the field, applied that fungicide product to see, are we losing any control in the field for these isolates that we're seeing a little bit of creep towards, less sensitivity. And essentially, thankfully, the outcome was we could still see really good inhibition, or really good control rather, where we sprayed with metconazole or tebuconazole. So that was really reassuring to see. We also looked at that SDHI fungicide. So this would be that other active ingredient in Mirabis ACE, so pideflumethafen. We went through and did a baseline sensitivity to get a sense of where are we at today. So these values here representing, again, those number of isolates and how sensitive they were to fungicides, this fungicide. And thankfully, the bulk were quite sensitive to that SDHI fungicide. So good news there. We also looked at, is there any correlation in the sensitivity to these different fungicides? So this is looking across these different FRAC groups, or the modes of action. So this SDHI compared to tebuconazole, do we see a correlation? And we didn't, which was good. When we did look between the different DMI chemistry, so tebuconazole and prothioconazole, we did see correlation, but we kind of expect that, because when you're in that same mode of action group and you have resistance to one chemistry within that group, you're more than likely going to have resistance to others within that group, because it's all targeting the same mechanisms for fungal control. So very briefly then, we did see some significant differences in sensitivity between the species, the Fusarium species that we looked at in vitro. And we'd probably expect that based on the biology of these different species. They have slightly different ways to metabolize the fungicide products. That probably makes some sense. We didn't identify any practical resistance to the DMI fungicides. That's really good to see. But that was only within Michigan, within a certain set of isolates. So we want to expand that across the region now. And as well, that Fusarium graminearum is also very sensitive to that new group, the SDHI fungicide, where we screened essentially the Mirabis ACE component, pideflumethafen. So that's helpful to see, and really good baseline information to help us track any changes in fungicide sensitivity or resistance development into the future. So really quickly, here's a plug. We would very much like samples. So if you work at an elevator or have heavy levels of scab in your grain this year, please feel free to reach out to us. Alyssa is up in Delaware, and she is sort of handling the south and eastern seaboard. So we're receiving samples over the last couple of years, and for another couple of years to continue monitoring for fungicide resistance. I'm handling sort of the Midwest and out west side of the US. So if you have any scabbed samples, we very much appreciate that. So with that, Andrew, I'm done.
Thanks, Marty. And kind of just add on to what Marty just gave a plug for sample requests. We have land grant institutions or local extension offices that certainly can help with the facilitation of getting those samples to the right people. So just, it's great. I mean, the more samples we have, just like anything, the better idea we have what's going on across the US. Right now, Marty, I'm not seeing anything pop up in the question and answer box. But, you know, I want to thank you for you and Alyssa really kind of taking this, you know, this effort to the next level. And I mean, could you give us any perspective on like the next steps of what we have for isolates, or how many you guys are currently handling across the US?
Yeah, we've certainly got a few hundred. We haven't really had a bad scab epidemic. This is the way it works, right? You start a project and then there's no disease to work with, which is great for our growers. I'm sure, you know, the time will come and we'll have more scab across the region. So when that happens, we certainly want to jump on that and collect as many samples as we can. And there's multiple aspects to this, certainly looking at fungicide sensitivity or resistance, and that's really important to understand what products are working or not working, but also the species of Fusarium that are out there causing issues. We need to, you know, be on top of that. That's also a really important part.
Yeah, absolutely. Marty, I do have one question that we'll have time for to answer. The first one I have is, have you compared the EC50 values from your study with concentrations of active ingredients typically used in the field?
Yeah, that's a really good question. That comes up all the time. So, yes, we have. We think about this question a lot. We do a lot of work with seed-borne pathogens as well. So it's not necessarily a question of what's in the tank and the parts per million in the tank, but it's a question of what ends up on the head and how that is distributed around the head. So it becomes a little bit of a difficult thing to pin down the exact dosage. But what I can say is that when we do find these isolates that have a little bit less sensitivity to the fungicide, we wouldn't call it resistance yet. We're not completely freaked out. But when we do put those isolates back into the field and spray with a fungicide, we can still control them. So in that sense, there's correlation between what we do in the lab and the field.
Sure. And then the last question I have is there any interest in receiving samples from other countries?
Yeah, we could potentially do that. Yeah, I mean, certainly the focus is through the SCAB initiative, but we are interested in you know, bigger picture of things too. It is helpful, so we could potentially look at doing that. So feel free to reach out.
Yeah, absolutely. So at this time, we're just going to take a quick 5-minute break, get up, grab some more coffee, and then the final portion for probably the last half hour or so is that we're going to invite everybody else back in and include a few new members. And we're just going to have what I call panel discussion. So during this break time in 5 to 10 minutes, please continue to submit questions. If not, I do have some material that I can at least start some conversations on frequently asked questions received as specialists. So again, 5 to 10 minute break here. Well, let's say we're going to meet back here at the 27-minute past the hour. So about a 6-minute break here. So with that, we'll see you in a few minutes. Welcome back. So the last portion of Scabinar today is I'm going to invite all of our speakers back into this room, and really want to have some discussion and conversation about maybe some of the questions that either were asked in the question and answer box or some of the ones that we can generate. So I would ask the presenters to please turn on their screens again. And at that time, we also have a few new members joining us on this panel, and I will take a moment to introduce them as well. So we have two other individuals in this panel that have joined us. First one I'm going to introduce is Dr. Juliette Marshall. She's the department head, professor, and extension specialist at the Department of Plant Sciences at the University of Idaho. And she has a strong applied research program focused on developing variety, production, and disease management information on small grain crops. And the other individual we have is Dr. Kelsey Anderson Onofre, who is an assistant professor and extension plant pathologist at Kansas State University. And her extension research program looks at basic and applied tools to answer critical questions in wheat disease management. And to kind of kick things off, I think this might be a good opportunity given the different regions for Juliette and Kelsey. Juliette, could you provide some opening statements on Fusarium head blight or scab challenges you have in Idaho or maybe in the PNW?
So we've been seeing a lot more Fusarium head blight or scab in both wheat and barley, mostly visual symptoms in wheat. Some of the DON has been accumulating in the barley because the visual symptoms aren't as easy to necessarily see in the field. A lot of this has to do with changing crop rotations. We're getting a lot more corn in the area. In general, it is directly related to the rotation associated with corn production. And our risk is directly related to that and less so to maybe wheat after wheat or barley after barley. So our control recommendations tend to focus on crop rotation, making sure that if grain is planted after wheat or if wheat or barley is planted after corn, then we focus a lot more on the increased risk. So due to the overall conditions, we have hot, dry winds and hot, dry summers. The overall conditions tend to really dampen the incidence of head blight in our area, except for in certain summers and increasingly under center pivot irrigation where we have constant irrigation going. So our risk is going to be a lot lower overall, and I think our control methodologies are going to be more effective overall because of that decreased risk and overall environmental conditions.
It's a very unique perspective. You know, you start talking about conditions, right? We know that drives scab epidemics, and irrigation is another component of how do we manage it. And so it's really, really interesting to see some of your perspectives. Kind of compare that a little bit more. Kelsey, down in Kansas or maybe in the Great Plains, how do you view scab management or your perspective on managing scab?
Yeah, thank you, Andrew. Yeah, so here in Kansas, I think similar to what Juliette mentioned, scab has been an increasing concern. So if we look back at historical yield loss trends, we really started to kick up in frequency around 2015, and we've had more frequent epidemics since that time. Similar to what Juliette mentioned, a lot of that trends with increasing corn acreage, right? Corn hybrids that are better adapted to our dryland conditions have been introduced on the market. So we just have more corn interfacing with wheat, right? Kansas is the #1 producer of wheat most years. Sorry, Andrew. Sometimes we jockey back and forth with North Dakota, but we have definitely are the biggest producers of red wheat, hard red winter wheat. With about 7 to 10 million acres per year, but we have about 6 million acres of corn, right? So that's a pretty large source of inoculum that we have in the state, and something we're pretty concerned about. Kansas does follow a moisture gradient, right? So eastern Kansas typically has higher moisture. In that part of the state, we typically do see scab more. We expect more frequent outbreaks in the eastern part of the state. One thing that was interesting this year, or last year, really in 2023, is that we had a pretty big outbreak in the northwest corner of the state, and that extended into Colorado. And that part of the state was not prepared. There probably hasn't been an outbreak like that in that part of the state ever. So there's been a lot of education and some learning that we've had from that experience there. We had pretty high DON levels in some isolated pockets, and that really was just high levels of corn residue. We've had bad corn stalk rot in the year prior, and and we just had those timely rains right at flowering, cool, wet weather. So it was a perfect storm, and something we're still thinking through and working through some education there. So thanks for including us, Kansas, in the talk here.
Yeah, I just kind of just jump off what Kelsey said. It's, all of us who's ever on this call today, or any land grad or extension specialist, they're going to have their story. They know where they're going to see scab, a lot of dynamics. We know the influence of corn, and we all can have that really bad scab year, or we may have several years of not seeing as much scab. It's just, that sporadic type of disease. But the thing I always remind people, it just doesn't go away, right? It's an ultimate survivalist. So thank you, Kelsey and Juliet, for joining us, and as well as the other speakers from today. And we have some questions rolling in, and I think we'll just start probably taking this off as they come in for the group. Feel free to jump in. We'll try our best not to talk over each other or have the awkward dance of who's going to talk first. But if that occurs, I'll provide some more direction on it. So the first one we have is, do any of the FHB fungicides have an effect on germination of the seed if you use it for planting next year? Carl.
Yeah, Sandra, I can jump in on that, I guess. So I'm not aware of any. I think as long as you're following the label, being aware of those pre-harvest intervals and applying according to the label, that I'm not aware of any issues that would come up because of a fungicide application on seed germination.
If anything, seed treatment of scabby seed can help with stand establishment. So the opposite here. We've done some work deliberately mixing seed, a different proportion of scabby seed with healthy seed, and treating those with a fungicide. I think the base fungicide at the time had tebuconazole. It's been a few years now, and the germination, the stand establishment was better with the treated seed at every level of scabby seed that we tested. So if anything, it's the opposite. If you have scabby seed that you want to replant, obviously, if it's too scabby, then you probably shouldn't replant it. But if you have to replant a scabby seed, I think the highest level we looked at is 15% seed treatment would help, especially seed treatment that has tebuconazole as a base product.
And if you're switching market classes, kind of building on what Carl and Pierce are saying, I'm not aware of any phytotoxic effects from that Fusarium head blight application in season, and for those that save seed for next year. And there's probably plenty of it, so we have just as much farmer conversation as we probably have research data on that. So thanks for the question on that. My next one, and this is a really good question. I'm gonna probably open this up a little bit more, but the question is, have you conducted spray trials with various resistant genes in wheat, such as FHB1? So I think the bigger question is, how have we conducted trials? Maybe the history and what we've done. Maybe Pierce, you want to touch on like the integrated management trials and maybe some of the objectives and what we have tried to establish over the last decade or so?
You want to give me, I'm going to share a quick slide here to help answer that question. Can you guys see that slide?
Hey, we can, Pierce.
Yeah, so we've pretty much done integrated management trials, just like we did with the uniform fungicide trials. We've looked at trials across multiple location years and market classes and compare some of the same fungicides I talked about earlier in terms of their efficacy on resistant and susceptible materials. And if you can look at this particular slide here, MR means moderately resistant, and this is across location years and market class. MS means moderately susceptible. Some people call this intermediate across locations and market class, and susceptible. And if you look at the data, just the course we look here, we're going to have, I think Amber's probably posted some of this information already on the SCAB website, so you can read there for more details. But these fungicides tend to be more consistent in terms of their efficacy when applied to a moderately resistant and moderately susceptible variety compared to when applied to a susceptible variety. If you can see, there's some variation here in terms of efficacy on the susceptible variety, but the lines are pretty flat and the letters are pretty much the same across fungicides when you're treating a moderately resistant and moderately susceptible variety. And that's why, even though I focused my presentation today on fungicides, and those were all fungicide efficacy trials on susceptible variety, we typically preach as a group the best way to control scab is combine the best fungicide with the best resistant variety. And when you're using resistant varieties, MS or MR, your choice of fungicide is, I wouldn't say less, it's less important in a sense because the efficacy is consistent. But when you look at the susceptible variety, then there's variations here. Here is number 2 here in this case is Miravis Ace. Number 1 here is Prozaro, and then here is FedEx and Prozaro Pro. So fungicide seems to make a difference more when you're treating susceptible variety, but when you're working with resistant or moderately resistant, you get best results with your fungicides.
Christina, you have anything to add?
Sorry, Andrew, were you calling on me?
Yeah, I don't know if you had anything to add as
Yeah,
far
yeah,
as
I just,
barley side of things. Yep.
I thought this might be a good occasion just to mention, since our session today is about fungicides, and that's been very informative and important, just a plug for the risk tool as the season gets closer, so that we're remembering that all this fungicide information is kind of against the backdrop of, do we have scab risk or not? And I think that research shows that even susceptible varieties, it's hard to profit from a scab targeted fungicide if there's no scab. So we need to be monitoring that risk and knowing our variety resistance levels, but targeting our fungicides if we have scab risk and if we have a moderately susceptible or susceptible variety.
Yeah, that's a great plug too. And kind of summarizing some of the insights is, using your best resistant variety with the fungicide is going to give you the most amount of scab control. It doesn't make it zero, but the more tools we add, the better off we are. And regardless of what market class you are, it's kind of the same story on that. Next question I have is there any correlation between Fusarium chemotype and fungicide efficacy? I remember some of this work was done before, but I hate to say it might have been done before my time, even of my familiarity. Don't mean to point fingers at some people in the room that might know, but I do know, I believe it was explored at some time. Does anybody have any summary statements off the top of their head?
Andrew, I can address it from the lab. I don't know if some of the more senior members want to speak up. But anyway, from what was done before, but from what we did in the lab, what was interesting, I guess, is that at least across Michigan, 15A DON predominated across the region. 3A DON and NX2 were more prevalent in areas near wild lands, essentially, for lack of a better term. And I think that's something that Gary was seeing over in New York as well, Gary Bergstrom. But we didn't detect any differences in sensitivity based in our lab assays, at least.
That's kind of what I remember from the story too. So kind of what Marty, you mentioned is, I'm not familiar with anything that would deviate from some of your comments, but there is some research looking at it.
Yeah, I'll happily wear the senior guy cap here, Andrew, in saying that I've seen some work from North Dakota. Don't remember off the top of my head, but what I do recall is the 3A DON tend to produce more DON than the 15A DON, but relative proportion of reduction compared to the check with fungicide is comparable. So I don't know that fungicide that it makes any difference in terms of the fungicide applied, but the fact that 3A Don tends to produce more Don than 15A Don in regions where it's prevalent means that the absolute value is higher, but the relative proportion in terms of reduction is similar. That's what I do recall seeing as a senior guy.
Yeah, but yeah, you're exactly right, Pierce. That's a good way of summarizing it. As far as some of the work that was done previously, especially looking at 3A and 15A Don and their impact. And I think it's a great way to capture some of the message on it. And then Marty, of course, giving some of the different viewpoints too on some of these other mycotoxin profiles that are associated with Fusarium. The next one pertains to soft wheats, and the question is, soft wheats have, since I'm not a soft wheat guy, pin genes, you're gonna have to correct me on this, that confirm some fungal protection. Have you compared the efficacy of fungicides in soft wheat versus hard wheat?
So I think Pierce is probably the one that is going to have the best answer since he has the data. But
Yeah,
I haven't said anything for a minute, so I'll say something. I do know I'm not sure what pin genes are personally, but I do know that sometimes at least in soft red, that there may be Dr. Fred Kolb, who used to be the breeder at University of Illinois, would kind of call those native genes, native resistance, and so that may be what Dale's referring to there, but. But Pierce, I think you probably have the data set that has kind of looked at least regionally, some differences in efficacy. So there you go.
Okay. Again, I've got buckets load of data, as you guys know, and I can't take credit for this one. This is my partner in crime, Larry Madden, summarized this here, but the red is winter wheat and the blue is spring wheat. And across those same fungicides I talked to early on, the early half of my presentation, the trends are pretty much the same, but the percent control in terms of efficacy seems to vary by region. This is not only now. The first set of data we looked at way back in the early 2000s show very similar trend. The trends are the same in terms of the products, but there tends to be a difference in efficacy from one growing region to the next. And if you look at this data, I'm not going to get into details here because we're still working on this, still trying to make sense of some of this, but it shows the trend that there is variation. And we argue sometimes, like Andrew raised a good point about anthesis window, whether anthesis window is in the spring versus anthesis window in the summer. And some of those factors we use as justification to explain some of these differences. But again, this is only speculation, anecdotal information as to why we're seeing these variations. Andrew can argue that fungicides have been used longer in the spring wheat areas than winter wheat areas as well, but we're not gonna speculate beyond this point.
I actually appreciate, Pierce, you have all this stuff ready to show a screen, ready to share on your screen. So it's almost like you expect these questions sometimes, or maybe you're getting insider knowledge. I'm not quite sure. But...
You didn't prime me? Are you sure?
I don't know. Maybe
no, I'm just kidding. I got this data in front of me here. We've just finished our annual report for the SCAB forum, so I've got this all somewhere on my desk.
Oh, I appreciate sharing that. It tells a pretty good story as far as some of the things we've looked at over the years. Moving along here, this might be a good one for Juliet, but the colleagues up in the PNW, they're starting to see an increased FHB pressure, and the question is, what do you think PNW research that can be done to best support growers to handle some of their FHB problems in the PNW?
At the University of Idaho down at Aberdeen at a research station down there, we've been addressing a lot of the questions regarding control of FHB, not necessarily in rotation, but under our high elevation desert type conditions, but under irrigation. And that's where we're seeing the biggest increase in FHB. And the conditions that we have in Idaho in these high elevation deserts are going to be similar in Montana and Washington and sometimes Oregon when we start talking about the environment. And again, in general, I mean, you could talk about Western Oregon versus Eastern, and same with Washington, but in these higher elevation areas, and or our desert type environments, the research that we have in Idaho should apply pretty broadly to a lot of these similar environments in Idaho, Montana, Washington, and Oregon. And I think that our conclusions can be broadly applied to the PNW. Again, especially as I mentioned earlier, when it comes to the heat and the humidity characteristics, which high heat sometimes, low humidity, but the integration and the simple fact that corn is increasing in a lot of areas due to increased adaptation, as Kelsey mentioned, of the corn into multiple environments, as well as the fact that the profitability of corn is undeniable, and that increases the growth. I know that our forecasted corn acreage in Southeast Idaho for next season is supposed to be pretty high. So that's going to have implications throughout the region as far as the increased risk associated with the production of corn and then following wheat and barley. I think another potential problem that we're seeing in our Pacific Northwest environment is an increased amount of unpredictable late season precipitation that is actually not only causing sprout damage, but is increasing our potential risk associated with some late season head blight. I am very concerned about the increasing potential for head blight to develop with increased humidity and increased precipitation events occurring in the wrong season, closer to anthesis or even late in past anthesis into some grain fill. But I do think that a lot of our research and the research results can be applied broadly across the whole PNW region and the Intermountain West.
Yeah,
Thanks,
that's great,
Andrew.
Julia. I mean, definitely, find the foundation for, we've talked about a little bit before, but the different climate, the high desert, the irrigation, all those factors, and certainly the tools are there. It's just, finding the research to fit the management profile. And it's interesting, Julia, and I just try to include as many panel members as possible, but you mentioned the influence of corn acreage. Kelsey mentioned the influence of corn acreage, but another crop that sometimes, not every state gets the question, but maybe specific to Kansas and some of that region down there is sorghum. Kelsey, do you have any information on what's the influence of like sorghum when it comes to rotation and the risk it has with Fusarium head blight?
Well, I can make a couple comments and then I'd be curious if anybody else on the panel has some information. So yep, Kansas is the #1 producer of grain sorghum in the country. So it is an important rotational crop for our wheat growers here. That corn, that sorghum wheat rotation is important, right? And so the same pathogen that causes Fusarium head blight causes a sorghum stalk rot, right? And some sorghum head diseases as well. So generally from the limited data that I've seen, we consider it somewhere in between soybeans and corn as an effect as a rotational crop, right? So from what I've seen, we believe that it is, surviving in that sorghum residue, but of course your corn is going to be your absolute highest risk, maybe followed by sorghum, and then, some soybeans or wheat in the rotation would be of concern. So, corn is again the highest risk, but we don't want to forget about sorghum. As you mentioned, Andrew, Fusarium graminearum is a great pathogen that loves to survive against all odds. So getting it out of a rotational system, especially when we have no-till, is very difficult. Again, I'd be curious to hear others' opinions on that.
Yeah, I don't see sorghum much. I mean, honestly, the only time up in North Dakota I see sorghum is usually in pheasant hunting plots, tell you the truth. So it's used for wildlife more than anything. So it's, I never thought of it in that dynamic, but certainly the host range and seeing how that influences from a management perspective. It's just, these are some of the same questions, regardless of what crop it may be, but an understanding of the survivalist nature of Fusarium, and it's not always created equal for a Fusarium host. And I think that's kind of been evident with some of the conversation. Just because I promise I'm getting every panel member involved. The one person I haven't heard from yet is Rob. And I think there's one question that I think would be really good for him to at least touch on. And oftentimes when it comes to making a fungicide application, you did a great job talking about the ground application. What about aerial applications? Do you have any perspective of if aerial applications work, or are they work when you need them, or any insight on that?
Sure. I haven't had a chance to look at aerial applications near as much as ground, but I did right around the same time that NDSU Extension published that ground application resource. There was an aerial application one as well, and I would say that it is certainly commercially viable to spray fungicides for FHB by air, and it just comes down to some of those application procedures. That document, it's essentially archived now. I should get around to reviewing it and maybe getting it re-upped, but I think the information within it is still good, and just some of the high points are, the spray quality is similar, that kind of median of 300 to 350 microns, which would be right around a core spray quality. The rotary atomizers on an airplane probably give you a little bit narrower span than a ground nozzle, which you might be able to get a little bit of a tighter droplet size distribution. But at least 5 gallons per acre is another, I think, key point. Aerial applicators for a lot of other types of applications are usually made at 2 to 3 gallons per acre, but really that 5 gallons per acre, a little bit higher water volume, is going to be important for scab applications. And then it's just making sure that your applicator has a good pattern tested airplane, that they know that their droplet size distribution, they know they're not getting a lot of driftable fines. They've done that work to get the plane set up properly, and you should have success with an airplane.
Rob, I have a question for you. I thought that the research had shown that aerial applications produce on average lesser coverage of the head than ground applications. Obviously you can cover a lot more territory from an airplane, and under certain conditions you can get in where you couldn't get in by ground when it's too wet. But is that not what the data show?
I'm not familiar enough with the data to say one way or the other. That very well could be. I think that, there's more, like, to compare apples to apples, I don't feel comfortable making a statement as to whether one is better than the other. It's if it's, but like you alluded to, reality sometimes dictate that you would prefer an aerial application versus a ground, especially if conditions are such that you can't get a ground sprayer in. So yeah, I spent a little bit of time going through that old extension publication, anticipating that I might get the question. And it doesn't really isn't any information in there about the efficacy of an aerial versus a ground. But there is a way to configure that aerial application to get as much success as possible if that's a route that someone is needing to go.
Mm-hmm.
I think Hooker in Canada did a lot of work on that previously and found that the actual coverage was much more effective with a ground application. And so that potentially would be similar to other areas. But it's kind of interesting. One of the growers and applicators that I've been working with in the past has sworn up and down that he could get just as effective coverage using an aerial application that he can for a ground rig. However, you also have to look at the overall environment, and we go back to the actual pressure overall in our environment, and the aerial application and some of the maybe different fungicides, not the top end fungicides, will be just as effective in our environment at reducing and preventing the infection, simply because that the overall disease pressure is a lot less.
And I guess two other points. One is that, that timing is really important. So, properly timed aerial application versus a poorly timed ground is probably still a better option, because that, I would say, timing is going to override your application parameters. And one thing for folks to be aware of is that spray drones are becoming a little bit more of a topic, especially under smaller field sizes. And there's some folks doing a little bit of work specific to application procedures with those, but just one general statement I can make is that be aware that the spray from a rotary spray drone is fundamentally different than an airplane. An airplane, it falls down as an even sheet onto the crop. Those rotary drones have a lot of turbulence that's actually driving the spray down into the canopy. So rules for an aerial application with an airplane are going to be different, most likely, compared to those with the spray drone, just because it is a fundamentally different approach of delivering those nozzles to the, or delivering those droplets to the canopy.
Yeah, Rob, you made a good, a comment about the use of drones, and that's something as a group we've had a lot of conversations apart in the SCAB initiative, and I know there's some folks that have been at least dabbling in it, and like anything, we grow with research and we grow with the data, and we continue to try our best to get that communicated out there. So we have a few minutes left. So I guess we'll take one more question from the question and answer live here, and then going to ask my colleagues here to see if they can address some of the other ones through the text response. And whatever we don't get to, we'll try our best to continue to answering them as we run up on time here. So I guess the last question for this group live I have is, have you seen any protection to test weight when wheat has had a fungicide but was harvested late? So I think maybe the situational awareness on this is supply of fungicide for scab, really delayed wet harvest. Are you able to protect test weight associated with that, or any type of comments related to fungicide being used to protect test weight?
I would say, and others can chime in, that the effect of the fungicide applied for scab around flowering will have pretty much dissipated and gone away by the time that harvest rolls around, and that's of course by design because of pre-harvest intervals being important, and that the impact on test weight from a late harvest is fundamentally going to be governed by whether there's rain, and of course every rain is bad for test weight, and how late the harvest is going, and whether it's rained are going to be your primary factors. I don't think the fungicide is going to have much of an impact.
I'd say most of my test weight responses have been, more on the disease control side. And it's kind of been independent of a wet or delayed harvest. So if you're protecting against a disease that's going to impact some of the test weight, that's when I've seen the biggest response, and especially with scab. If you had some scabby years, for sure, you're going to see a test weight response. Almost all that has to be doing with fungicides to use to manage it. So again, I just I promise to make this a 2-hour Scabinar, so I'm always going to do my best to follow by the time. I encourage the panelists to keep on answering questions as they are still coming in here. We have a couple left, but on the screen here, for those that are interested in for this training for CCA, CEU credits, the first screen here is definitely the QR code that can be used. The second thing that I kindly ask is, we listen to the feedback, we listen to the messages that we received in 2022 from our Scabinar, and we ask you, if you can, please fill out a survey. I know everybody always submits a survey, but this is really helpful for us to know how we did today, but also to see what we can do for the future if we have another Scabinar event. So again, another QR code that you can use to access that survey and provide your honest feedback for us. That's how we best learn. So as we kind of wrap things up here, again, want to thank you for your attention today, chiming in. This is recorded, so if you ever want to go back and review some of the information, discussion topics, certainly you can do that. Also, you have excellent land grant and extension resources in your areas. I advise you to work with them if you're looking to get information specifically and connecting with the greater network of pathologists we have working on scab in the U.S. And finally, last round of thanks again for the U.S. Wheat and Barley Scab Initiative. And I also want to thank some of the valuable members in the Scab Initiative office of Michelle Birkness and Amber Hofstetter, as they're still working behind the scenes making this all up and running. So I appreciate that. And then finally, my colleagues, thanks for giving the presentations today and entertaining discussion, and look forward to see what next year holds for us.